Okay, welcome. This is the third lecture. This is an introduction to eukaryotic cells. So let's move forward. Eukaryotes, they can be unicellular or multicellular. They all have a membrane-bound nucleus. They have membrane-bound organelles. Some are pathogenic, so they're pathogens, some of them. These include fungi, animals, particularly in the group of helminths, plants, and these can produce harmful toxins, and protists. Now, how did we get from prokaryotes to eukaryotes? The endosymbiotic theory tries to help explain this. This is an explanation, an attempt to explain the origin of eukaryotes as a series of sequential cell-merging events between ancient eukaryotic ancestor and certain prokaryotes. Now, an ancient non-photosynthetic prokaryote was engulfed by a larger protokaryote. And what you have is the development of the mitochondria. Now, why do we say this? Because mitochondria have a distinct DNA, ribosomes, and they divide like binary fission, just like bacteria. When I say distinct DNA, they have a circular loop of DNA as opposed to the eukaryotic cell, which has it more a linear strand. Now later on, it's believed that a certain photosynthetic bacterium was engulfed by a primitive proto-eukaryotic cell. The one thing that chloroplasts and mitochondria have alike is that they have the electron transport chain found in the plasma membrane around organelles. They have DNA in a single circular loop. The ribosomes are similar to bacteria with a basic sedimentation rate of 70. The organelles split by binary fission. Now, to help you, yes, mitochondria basically are very important in the cell in that they will produce a lot more ATP per molecule of glucose. Chloroplasts take light energy to help synthesize glucose. But let's take a look at some of the other things that are important here. When they did gene analysis, they cites a close relationship between mitochondria with the rickettsia bacteria. Rickettsia are a unique bacteria that basically have to really be inside the cell to really function. The chloroplast has been found to have a similarity genetic-wise with the cyanobacteria. So what we have here is sort of, and when we talk about symbiosis, that's really the process of cells or organisms living together. So we have this formation here over time, and what we're talking about is not 1 million or 2 million, and we're talking about billions of years ago. And one proto-eukaryote was starting to evolve a nucleus, a protective membrane around its chromosome structure. But meanwhile, it engulfed an ancient non-photosynthetic prokaryote, maybe something in the Rickettsia species. And as it engulfed it, instead of consuming it, they set up a living arrangement. Larger cell provides shelter and nutrients for the smaller cell. Smaller cell is able to extract out and produce many more ATPs per molecule of glucose. If you just go in this particular direction for now a couple of billion years, you end up with the precursor to animals, fungi, and non-plant-like protists. So where did the plants come in? Well, what happened is one of these organisms instead had another situation where a photosynthetic bacterium, perhaps cyanobacteria, was engulfed, and in this case, it was sustained. And the chloroplast developed, and now you had the capability to have a primitive photosynthetic eukaryote. And this may be the precursor for plants and plant-like protists. Okay? So we move forward from here, And we note that there are some developmental differences between prokaryotes and eukaryotes. First off, eukaryotes have a membrane-bound organelles. Their DNA is sequestered in a membrane-bound nucleus. The ribosomes are different in size and location. Some of them are free-floating. Others are attached to the rough endoplasmic reticulum. The chromosomes aren't circular, as what you see in prokaryotes. Rather, the eukaryotes have chromosomes that are linear. The cell walls may exist in plants, fungi, and select protists, but not in animals. The cell membrane of eukaryotes contains sterols. An example would be cholesterol, and that's not found in prokaryotes. The cell division for eukaryotes includes mitosis, which would be an asexual means. You start with a cell and you end up with two daughter cells genetically alike, or meiosis, which is a process that is considered sexual, and you end up with genetic variations which allow for enhanced fitness and survival, especially in a environment where you have changes coming from time to time. The cell size is also much, much larger than prokaryotes. Okay. So if you take a look here, we have the eukaryotic versus prokaryotic. I encourage you to take the time to review them. But the key points here are, look, with organisms, eukaryotes can either be unicellular, such as protists, or a type of fungi, the yeast, the single-celled one. Multicellular, they can be plants, animals, and most fungi. When you look at prokaryotes, they're all unicellular, and they're either from archaea or bacteria. The size comparison is that eukaryotes are usually much larger than prokaryotes. For cell division, it's either asexual or sexual, whereas in prokaryotes, it's always asexual and it's the form of binary fission. In the plasma membrane, eukaryotes often contain some type of sterols, yet prokaryotes, it's rarely found to have sterols. Cell wall, yeah, some prokaryotes have it, plants, fungi, certain protists for the eukaryotes, but for prokaryotes, most of them have it. The only exceptions are in the mycoplasma or in the L forms that are pleomorphic. Nucleus, yep, exists in the eukaryotes, but not in the prokaryotes. Ribosomes. The ribosomes in eukaryotes are ADS for the cytoplasmic ones, the rough endoplasmic reticulum. But remember, if you have either mitochondria or chloroplast, your eukaryotes are going to be 70S, just like the bacteria. 70S. The genetic material for both is the same, DNA. The chromosomes, though, may be multiple linear chromosomes for eukaryotes, whereas in prokaryotes, usually you have just a single circular chromosome. Okay, here is a kind of generic type of eukaryotic cell. You'll note the plasma membrane. On the exterior here of it, it'll be a glycocalyx. You'll have rough endoplasmic reticulum with the little ribosomes there. There may be free-floating ribosomes around too. You're going to have peroxisomes. You're going to have the Golgi apparatus. You're going to have smooth endoplasmic reticulum. And, of course, mitochondria here, 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 whatever. You'll also have a cytoskeleton and a centrosome, not to mention also the lysosome. And on the exterior, you can have a flagellum. Now, cell division. Quickly, we will go over this. In the prokaryotes, it's always binary fission. Eukaryotes have two options. One, an asexual reproduction of the cell division, basically mitosis. You end up generating genetically identical offspring from the original mother cell. For example, all human cells, except gametes, divide by mitosis. Now, with meiosis, though, this is where you make gametes, in other words, haploid cells. For example, in humans, you have sperm and egg. And when they combine, this is fertilization. We have the formation of a zygote. A new genetic combination occurs. In fungi, they don't use the term gametes, sperm and egg. They use the term gametes in the sense of spores. The spores are formed, and they combine to create a new diploid cell, a zygote, with new genetic composition. I encourage you to take time to review this chart here, but as you can see, here we start off with mitosis, a diploid parent cell, DNA is copied, cell division, 2 diploid daughter cells. Meiosis is different. You have the process where you start off with diploid parent cell, but the DNA is copied, and then you have these reductive states where you don't have the time to make more DNA, so you go from diploid to haploid daughter cells. Now, haploid means you have 1/2 of the number of chromosomes that normally a diploid would have. Humans would be 46, and that's diploid. Haploid would be 23. But when you have these 2 haploid cells fused together to form a zygote, you now reestablish yourself in a diploid status. Prokaryotes, and don't forget mitochondria and chloroplasts. Binary fissions, basically asexual reproduction. You copy the chromosome, the cell divides, you have exact copy. Now, from there, we're going to move into cell transport. This is where we talk about briefly exocytosis, endocytosis. Endocytosis is the process to bring substances into the cell. Exocytosis is the process to transport substances out of the cell. Both use ATP for energy and require specialized receptors for transport. Now, the different types of endocytosis include pinocytosis, known as cell drinking, phagocytosis, which is referred to as cell eating. Basically, you engulf a solid matter, as opposed to pinocytosis, you're engulfing a amount of a liquid of one type or another. Phagocytosis is well known. We see this with immune cells, with amoeba, and other protists that engulf the cells. When you engulf something, and it's now encapsulated in sort of a membrane-bound vesicle, it's called a phagocyte. Excuse me, it's a phagosome. A phagocyte engulfs the cell into a phagosome. Now, a lysosome is an organelle that contains hydrolytic enzymes. In other words, enzymes that break down substances, proteins to amino acids, lipids to fatty acids, polysaccharides into monosaccharides. What happens with certain phagosomes is that the lysosome will be fused to it, making a phagolysosome. When this happens, you will basically break down whatever is trapped inside the phagosome into its basic substances. Those substances then may either be expelled from the cell or reused. Now, you have to note something else. Some pathogens can escape phagocytosis and exit the immune cell. We'll get more into this when we get into host microbe interactions later on in the course. So here we have endocytosis. Something binds on, is engulfed, forms a vesicle here. Exocytosis is when you have a vesicle filled with certain substances, the vesicle fuses with the cell membrane, and the substances are expelled out. In this case, what you have is a target, which when engulfed has a phagosome. Here's a lysosome with its hydrolytic enzymes. These fuse together, forming a phagolysosome. The enzymes break down the substances that's in this phagosome, or phagolysosome now, and then it may be either expelled or broken down to substances that can be used by the cell. Okay? Here's an example of a scanning electron micrograph of a human neutrophil engulfing Staphylococcus aureus bacteria. Now, there's this particular type of endocytosis called receptor-mediated endocytosis. Now, a target substance that is to be brought in is called a ligand. The ligands will bind to receptors that are on the cell surface, and then they undergo endocytosis. Then we have a process called clathrin-mediated endocytosis. Now, this is an addition to the receptor-mediated endocytosis because upon receptor-ligand binding, the cytoplasmic side of the cell becomes coated with the protein clathrin, which will polymerize and cause a pit to form. So it's sort of like a sinking crater. The clathrin-coated pit draws in the receptor-ligand complexes, pinches off, and becomes a clathrin-coated vesicle. Now, the vesicle may combine with an endosome, which is a small vesicle with an acidic interior. This will cause the release of the ligand from the receptor. the receptors can then be recycled because they'll be sent back to the cell plasma membrane. Or you will have a lysosome that will break down both ligand and receptor during the hydrolysis process. Here you have this process. Here's the clathrin, here's the receptor, the ligand. You have enough of them, you form the clathrin, you form this clathrin-coated vesicle. The clathrin eventually will break away, okay? It will be shed off, and either an acidic endosome fuses with it. You release your ligand, the receptor's still good, the membrane material and the receptor attach back to the plasma membrane to be reused again. Or you have a lysosome that will attach and break down this ligand. Now, by the way, this can happen all together in several steps, okay? Now, exocytosis, this is where the vesicles deliver product outside of the cell. You see this with getting rid of waste, with expelling of hormones or neurotransmitters. As a matter of fact, with some other animals, such as snakes, where you have venomous snakes, this is how they basically release venom into their venom sac. The cells that line the venom sac will just basically, by exocytosis, release the venom into that by exocytosis. Now, exocytotic vesicles bud off from the Golgi apparatus. The Golgi apparatus is going to package this particular material, send it off on its way. Now, the vesicles may fuse with the cell membrane to release the products. The exocytosis is a process, as I said, to export waste, signaling factors, other proteins. Exocytosis vesicle membrane may be used also at the same time to replace cell membrane lost in endocytosis. Here we have the nucleus, here we have the Golgi body. We're starting to take our cargo here, concentrate it, and package it into a nice little vesicle that will then be sent, attached to the cell membrane, secrete the cargo, and this membrane here may add to the cell's plasma membrane. You can also have the process of both of them occurring. Let's say this is on the inside, let's say blood, and let's say this is just endothelial cell. And let's say you have extracellular fluid here. You would take, by endocytosis, put together some of the material that you want, transport it via this vesicle, fuse it with the plasma membrane on this side of the cell, and export it out. And of course, endocytosis is the reverse. You can bring products in. Now, we have to talk for a minute about eukaryotic organisms and the different types, and how some of them are health threats, etc. Eukaryotic organisms fall into 4 different kingdoms: animals, plants, fungi, protists. The animal kingdom is the largest of the kingdoms, but a particular subset of them is of key interest in microbiology, medical microbiology particularly, and these are the helminths, which consist of the cestodes, nematodes, and trematodes. Many of these are parasitic. What I mean by that is they live in or on the outside of the host, and they cause detriment to the host while the parasite itself benefits. Some parts of the life cycle of many of these type of parasites are in the gastrointestinal tract. Many of these have complex life cycles, including the use of several different host species. For example, there are some parasitic worms that start off as eggs, then go to another stage where they may infect an intermediate host, such as a snail or an isopod. Then they will go and be released and eventually move into humans. Overall, helminths are a very serious medical health burden worldwide. You can't go to any part of the world without dealing with some of the helminths in one way or another. Another part of the animal species are the arthropod species, because instead of them being a parasite themselves, they are vectors, that is, organisms that spread infectious agents to other hosts. There are two types of vectors. Mechanical vector, these are ones that physically carry the infectious agent to the host. Example, fly. On the feet of fly, they can fly from one site to the other, and on their feet may carry various types of bacteria. Okay. Cockroaches will have salmonella bacteria on their bodies, and so they will release this bacteria into places that you really wouldn't want them to, like a dining room dish set or something else like that. Yes, I know some people are curling over that one, but here's something further. A biological vector carries and or incubates the parasite inside their body. Then they transmit it to the new host, transmit that pathogen to them via feces or a bite, etc. Let me give you two types of arthropods. One class is called insects, another is arachnids. Insects have 3 body segments, arachnids have 2. We'll get more into this later on in a lab for vectors, but let me just give you a couple of examples to get you thinking. The trichomy bug is also known as the kissing bug, and this is a bug that bites individuals while they're sleeping. It takes in some of their blood. Now, the trichomy bug is also the carrier of trypanosomas that cause Chagas disease. The trichomy bug, when it takes in some of the fluid of the blood at the bite site, it has this hydrodynamic condition where basically fluid comes in, causes fluid to go out the other end. So you see it drop a fecal droplet that looks somewhat blood-like, but it will be rich in concentration of the trypanosoma. The individual wakes up the next day, scratches on around the eye or something else where the bite site is, and that will allow the parasites to go into the body. Let's not forget about the mosquito. The mosquito, interestingly enough, only female mosquitoes really bite. Why? Because the female mosquito needs a high-protein meal for her eggs that she will lay. But in taking in that high-protein meal, she becomes part of the life cycle of certain types of parasites, such as malaria. The malaria will basically reproduce in the gut of the mosquito, migrate to the salivary glands of the mosquito. When the mosquito takes another bite of someone else, they will always release some of their saliva, which acts as an anti-clotting agent, and in some cases will act to dull the sense of pain. And they immediately are, in essence, injecting some of the malaria parasites at that bite site. Okay? When we talk about arachnids, let me just give you one quickie. The flea. The flea, by its bite, is nasty, because what the flea will also carry is plague bacteria. Not all of them, but some of them. The flea has also been known to be a vector to a variety of other diseases, other microorganisms. The tick, equally so. For example, the tick, everybody gets concerned about ticks now because of Lyme disease. But other ticks will carry other types of diseases. Heartwater, they will carry also Rocky Mountain spotted fever, and a variety of other ones. So that's the difference between mechanical and biological vectors. And I would like you to just generally review these different types: animals, plants, fungi, protista. When we get into helminths, though, you do need to know these a little bit more in detail. Nematodes, otherwise known as roundworms, you have a variety of them. Pinworms, ascaris, filial worms, trichinella, strongyloides, whipworm, hookworm. Let me give you a couple of points. Hookworm bores in through the feet from soil, usually the soil somebody has defecated in, and so the hookworm will go in as its eggs hatch, and it will eventually get into the intestine. And in the intestine, as you can see that nasty teeth there, they bite into the sidewall and basically feed off the blood of the host from the blood vessels that are in the intestinal wall. Trichinella you probably all heard about how you're supposed to cook your pork until it's white, no red. Reason? It's been well established by FDA and USDA that they cannot prevent trichinella in the pork population. Hence, it is potential that all pork has trichinella in it, so you don't eat pork basically raw. Because if you did, you would get trichinella and they form incysts in the muscle structure. It's nasty. Let's go to flatworms. Two types, cestodes, trematodes. Let's deal with cestodes first. The best known are tapeworms. Now, this is an unusual species in the sense that it's hermaphroditic, which means that it has both male and female organs in not only the same individual, but in each one of these tape segments. Okay? And these will create the conditions where it can self-fertilize itself and produce a prodigious amount of eggs. These will occupy the gastrointestinal tract, the tapeworms, and they basically absorb the nutrients and they pass out their eggs in the feces. Where do tapeworms come from? Well, it depends if you've not cooked your beef, your pork, your fish. There's 6 different ones that infect humans. Okay? And when we get to flukes, these are trematodes. Now, trematodes, except for blood flukes, are all hermaphroditic, meaning that they have both male and female reproductive organs. In the blood flukes, such as schistosoma, as you can see here, this is the male, this is the female. What they will end up doing is laying their eggs and depositing them in the lower blood vessels near either the bladder, urinary bladder, or the intestine, and the eggs will be eventually deposited out into the feces or come out in the urine, and then they will eventually be reproduced again via different routes such as the oral fecal route, etc. They will, in some cases, the eggs hatch, and they will require an intermediate host. In this case, snails. Okay. Now, with the other types of flukes, which will infect and occupy the lungs, the liver, the intestine, you have these that will act and get into the body, usually by contaminated food or water. And what they will eventually do is cause a great deal of damage to these organs, and in many cases eventually kill the individual. Okay? All right. Let's talk about fungi for a few minutes. Most fungi organisms are multicellular. The only example of the exception to the rule is yeast. Yeast is the only single-celled fungi. Now, if you say yeast, what are we talking about? Well, we're talking about Saccharomyces cerevisiae, basically brewer's yeast, baker's yeast, you know, the stuff that makes bread, the stuff that makes wine, beer, things like that. Now, fungi are part of this group that is either a saprophyte or a parasite. Okay, saprophytes basically consume dead matter, you know, decaying leaves or breakdown of an animal body or something else like that, to get their nutrients, because they will release enzymes out of their cells that basically break down the dead matter, and they can consume that material, they absorb it via their body structures. Now, there are those that are parasitic, which consume tissue from a living organism. Now, with a lot of fungi, they have these structures, these tubular structures called hyphae. Now, the hyphae can be septate, that is, separated by walls, or aseptate, where there is no cell wall structure. That's basically the cell acts as a concentric, which means it has multiple nuclei. With a large cell cytoplasm, it's like one big, long, continuous tube. That's one big cell. Also, we have the concept of dimorphic ism, or dimorphic is this is a life cycle that consists of hyphae formation, then single-celled yeast formation. And this depends on what is the conditions of the environment for the fungi? An example of this would be, there's a type of fungi that is out in the southwest, particularly if it's dry, and they will act as yeast-like formations in the soil. And when the soil is blown up as dust, individuals that don't have the resistance, they breathe it in, and you start having the formation more from a hyphae approach. And you can see this in the brain and in the respiratory system, etc. So in other words, you get single-celled if it's a dry, hostile environment, and then if you get into a nice, moist, dark environment like the lung, you start sprouting out in a hyphae formation. So that's dimorphic conditions. Now here you see the septate hyphae. Here's the septate, the walls here, 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 here. Don't be fooled, because on the outside they all have a cell wall. Here and here is a cell wall. But the difference is that they have a septum, in other words, separation. So you have a single nucleus, separation, single nucleus, separation. Separation here, single nucleus. Now, with aseptate, no cell separations here, no septums. So now, in essence, you have this large continuous cell that has very large cytoplasm and also multiple nuclei that basically keep the entire organism functioning. When I talk about fungi, I have to talk about spores. Now, fungal spores, due to varied reproductive life cycles, you're going to have basically asexual spores and sexual spores. Asexual spores are arising from mitosis, and these types are called candidiaspores or sporangiospores. Sexual spores are the result of union of complementary mating strands of fungi generated by meiosis, which these are going to be haploid and then they will fuse. And when they fuse, you get genetic variation. These types include zygospores, ascospores, and basidiospores. Here's an example of the candidiospores. Here you can see this sort of strand coming off of this penicillium. And each one of these spores would break off and start a new fungal organism. Take a look here, a sporangiospore of the Absidia. This is almost like a flower top, or you might think of it like almost like an ice cream cone in its shape, but really what it is, this is containment membrane with lots and lots of these spores. So when this breaks, these spores are going to spread out and seed individual new Absidia. Now this differs from what you see over here, zygospores of Rhizopus. Now the Rhizopus will have basically a haploid spore and a haploid spore. They'll meet together and form this zygospore. This is now in essence a diploid organism, okay? And by the way, Rhizopus, if you haven't heard of that before, that's what you see in bread that has formed a mold, okay? Rhizopus is there's some Rhizopus species that basically are those ones that cause moldy bread. Now, ascospores. This is a particular type. This is a structure that you would call an ascus. It's almost like a tube that contains the ascospores here, 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 here. And these would eventually break off, and the ascospores would basically start new cup fungi, but they have genetic variations within them. Finally, we get to the basidiospores, and this is found commonly with mushrooms. You know, the mushroom spores that you see falling out here? Now, here is the basidium here, and here's the basidiospores right here that are formed, okay? And to help you here, what I want you to get away with is the asexual fungal spores, and these would be conidiospores and sporangiospores, okay? And you have the sexual fungal spores that are formed, and these include zygospores, ascospores, and basidiospores, okay? Now, fungi can do several different things to cause illness. One of them is they can directly cause diseases, and these are called mycosis. We'll get into several other ways in which they can make individuals sick. Many of the mycoses that we see now are with individuals with weakened immune systems. For example, it used to be called Pneumocystis carinii. It was thought to be protozoal. It was later found to be really the Pneumocystis jirovecii, which is a pulmonary infection, a fungal infection. It's very common in AIDS patients. Remember, with weakened immunity comes greater susceptibility to many organisms that we normally fight off with a healthy immune system. Dermatophytes infect skin, hair, nails. And what they do is they break down the keratin proteins, which are found in rich quantities in skin, hair, and nails. And then we have to deal with tinea, which is a common dermatophyte infection. Formerly it was called ringworm. Tinea infections are often referred to the site of the infection. So for example, tinea pedis, this is athlete's feet. And the fungi is a trichophyton. Now, aside of making you sick by infecting you, some fungi release toxins. And these toxins will cause a lot of illness. Mycotoxins are fungi-released toxins. If you talk about black mold in houses, you may have heard of black mold in houses where, either buildings or houses cannot be sold until you get rid of all the black mold. Sometimes they call it sick building syndrome. You would have situations where offices with suspended ceilings, the suspended ceiling tiles are made of some type of cellulose or some other material that a particular mold organism feeds on, but also releases the toxins. Black mold of toxins, the well-known organism is called Stachybotrys-chartatum. You need to know that. Also, Candida aureus. This is a fungal infection that affects immune-compromised patients and leads to infections of the bloodstream, wounds, and and ear. But here is the important point I want you to be aware of. This is different from Candida albicans, which would cause, yeast infections in women's reproductive areas, particularly the vagina. But the key point with Candida aureus is this. Right now, we're seeing drug resistance of this species, and it's spreading rapidly around the world. So this causes great concern because if you have a drug-resistant strain of some organism, it can really do damage to many individuals, and there would be nothing to do but basically provide palliative, that is, supportive care. Here's an example of Tinea ongonium, or dermatophytic onchomycosis. This is causing the discoloration of the nails. They become brittle and discolored. And this is a little bit different than, let's say, athlete's foot. Usually you see this a lot when you have individuals with nail damage, with immunosuppression, or they've been applying acrylic nails. They have to have that removed. And this is what you would see here. Now, athlete's foot, you can see what you have here is an SEM of fungal spores, the yellows, on the skin of a human foot, which is pink. Okay. We're going to get more into some of the ways to deal with this in the antimicrobial drug chapter. That'll be later on in the course. Let's move now forward into protists. Now, these include multicellular life forms such as algae. Now, this is important because algae used to be associated and people thought, hey, it's a plant. No, it's not. It's part of the protists. Protists, many of them are also single-celled life forms, such as slime molds. The single-celled best-known protozoan are slime molds, but there are a lot of other ones too. What we're going to focus in on is not the ones with plant-like characteristics, but more animal-like characteristics. Okay, so I want you to understand that. And so the characterization of the protists are going to be that they are animal-like, so they lack chloroplasts, they're unicellular, they lack a true cell wall, they exhibit both sexual and asexual reproduction, and they live by what is called heterotrophic means, okay? So they're not making their own food and obtaining energy via photosynthesis. Okay, so what are they going to do? They're going to consume other organisms for energy and organic compounds. This is important, okay? And they're not saprophytic, so it means that they're actually infecting or consuming live organisms or live organism tissue. Now, the ones that are pathogenic, this is where we're going to focus in on just 4 groups, and the 4 groups are characterized based on their motility, or it's how they move. We have the amoeboid, which is Sarcodinia, the flagellated, which is Mastigophora, the ciliated, which is Ciliophora, and the spore-forming, which is Acomplexia. Here's a photo just beforehand. When we talk about protists, it has turned out to be at times a grab bag for all these very unique life forms. But kelp are considered protists, even though they photosynthesize and they can grow tremendously large. Another interesting one is dog vomit slime mold. Yes, that's the name of it. I don't make this up. It's named for its texture and coloration. It's a lot of times you can see this in gardens. Now, why? Because if they have mulch, they have large amounts of, let's say, wood chips or something, boom, you're going to see this happen in later part of spring, beginning of summer. It just spreads out. Beginning, it looks more yellowish, and eventually it becomes more brownish red before it eventually hardens to a dry mass that's filled with spores. That's it. Now, let's start off with the protist, the amoeboid, also known as Sarcodinia. These use pseudopods for movement. So they basically have these extensions flowing outward of their cytoplasm, encased by, of course, the plasma membrane, and they move that way. Now, amoeba proteus, that's a safe form. You see that in high school laboratories and things like that, but there are dangerous ones. And yes, you need to know these. Naegleria fowleri is a very dangerous one. It has been found to go up through the nasal area into the brain. It's particularly found in warm waters in summertime. People plunge in head first. If these warm waters are not disturbed, so it's sort of like a pond or something else like that, but it's very warm, they will be ample. And if they get up into the nasal area, they travel up through the cribriform plate, infect the brain, and death usually occurs in 10 days. And there's not been many means to actually treat it. Acanthoamoeba, these are well known to cause eye infections, eye ulcerations. They occur when people have been having contact lenses on for too long. Balathumia, this is cause of encephalitis. The big one in Sarcodinia is Entamoeba histolytica. This is the cause for amoebic dysentery. Now, amoebic dysentery is a nasty situation. You have ulceration in the colon. The amoeba are there and they will flow in and out, feeding off of the blood, etc., but leading an individual to have hemorrhaging and dysentery, which would be a lot of diarrhea, etc. And it can be treated if it's detected in time. The flagellated protists, the Mastigophora, move by a flagella. The best known ones are Trichomonas vaginalis, which is a sexually transmitted disease. Trypanosoma, there's several types. The one from Africa causes sleeping sickness. The one that's called American trypanosomiasis, which is another name for Chagas disease, which I've talked with you about before. Finally, Giardia lymphoma. Now, giardiitis, you may be familiar with if you're a camper, because people will tell you, watch where you get your water from to drink. If the water, even if it's a stream, flowing from a beaver dam or a beaver den, don't use that water unless you treat it, because if you drink it straight out, you're going to get giardiitis. The beavers are a natural reservoir for Giardia lymphoma. Now, we move from there into the ciliated, the ciliophora. These use cilia for transport. A safe example of this would be the paramecium, and they have cilia moving along, pushing their bodies along. There is one type that's very significant, Balantium coli, which causes balantitis, which causes intestinal damage. Finally, we go to the spore-forming acomplexia. The spore-forming protists are the largest phylum of protists, and they move by gliding. They're an obligate intracellular parasite, which means that they have to be inside of the cell. They will undergo 3 stages of their life cycle: merigony, which is an asexual reproduction; gamagony, which is a sexual reproduction; and then sporogony, which is asexual. Usually at sporogony, these are the spores invade a new host to repeat the reproductive cycle. There are a variety of spore forming. I'm just going to take a few of them that are extremely important and quite notable. Toxoplasma gondii, which is causing toxoplasmosis. Toxoplasmosis is very well known as a disease. The reservoir for it happens to be cats, and you have a high chance of getting it if you are carelessly handling cat poop or emptying their little litter boxes. They're particularly important, it's particularly significant that toxoplasmosis, which eventually they get into the brain and cause abnormal behavior. It's also important to note that toxoplasmosis, there's a high risk of it for pregnant women if they are emptying the litter box. Because what happens is that they get the toxoplasmosis, the parasite will go in and either injure the developing fetus or kill the developing fetus. Okay. Now, Plasmodium species, it's another type of spore-forming acomplexia. Best known, malaria. The sexual phase of its reproduction is in the mosquitoes, as I mentioned before. The asexual phases occur in humans. And what it is that one stage will actually infect liver cells. The other phase basically will infect red blood cells. And as the spores build up in certain numbers and begin to release, they burst the cell. This is something that can cause eventually death because of loss of blood and damage to the liver. Cryptosporidium hominis, otherwise the pathogen that causes cryptosporidiosis. Now, this is an unusual one. If you think of the word crypto, it's kind of like something hidden away. The spores are in the water, and they can parasitize the intestinal wall epithelium. And here's a point. The mid-1990s, they started having outbreaks in large communities like Minnesota of cryptosporidium. What happened was they started to reduce the amount of necessary chlorine to treat the water for drinking water. And the cryptosporidium had built up some resistance to doses of chlorine. This led to a large city having a major attack of cryptosporidium, many cases. So what they did from that point on was boost the amount of chlorine and also use several other disinfecting strategies. I think ozone was one, and another was UV light to basically kill off the spores or these organisms. Now, by the way, when I say spores, spores and protists are far different from endospores in bacteria. The spores in the water, for cryptosporidium, for example, they don't have the resistance that endospores do. So it is possible to chemically destroy them, to using ultraviolet light to destroy them, etc. Okay? Now, here is an example of the amoeboid. You see the pseudopods here. This is Entamoeba histolytica. You can see Giardia limbilia with its flagella. And by the way, these two large areas that look almost like eyes, they're not. They're actually adhesion pads that the Giardia will attach to the intestinal wall. And with sufficient numbers of them, individuals start having diarrhea, malnutrition, etc., because they're literally Giardia coating the walls of the small intestine and blocking nutrient absorption. Here's an example of a ciliated one. This is relatively harmless, Tetrahymena thermophilia. You can see all the small hairs of the cilia, which help propel this cell-long. The last one is the spore forming, and here you see Plasmodium species in its sporozoite stage. Plasmodium, there are about 4 different types of Plasmodium species that cause malaria. Those individuals that are infected with multiple species at the same time do not have a very good prognosis for survival. And in some cases, I've heard of cases where they had all 4. And this was a soldier in Vietnam, and he died within like 4 days afterwards. So here you see the merigony, which is sexual reproduction. It will be in a host cell. The merozoites are released. The merozoites will eventually go into forming gametes, and the gametes fuse, and you have the zygote. The zygote will go through multiple repeated cell divisions to make sporozoites, and the sporozoites will infect host cells. Okay? Now, we're going to move into general characteristics that you see in many different eukaryotes, and we'll start with the extracellular structures. All eukaryotes have a plasma membrane. Yes, it consists of a phospholipid bilayer. Okay. They may contain sterols, such as cholesterol, to stiffen the membrane. Now, for animals, that sterol is going to be cholesterol. For plants, that's phytosterols. For fungi and protists, that's going to be ergosterols. The interesting thing is that if you have some biochemical differences and you know the synthesis steps for that particular chemical, in this case ergosterol, you can then begin to start examining and designing drugs that will block the enzymatic synthesis of the particular compound. And if it's essential, that drug can basically lead to the death of that pathogen. In this case, ergosterols, we find that azole drugs and other antifungals like amphotericin B and nystatin will target and block ergosterol synthesis and thereby treat and kill off the pathogen. Also, cell walls. These are used to protect against osmotic stress and maintain cell shape. Now, plants have their cell wall made of cellulose. Animals have no cell wall at all. Fungal cell walls are made of chitin. Protists, their cell walls vary. They can be either cellulose or calcium carbonate or xylan or silica, etc. Here we see the nucleus. This is a transmission electron micrographic photo. And what you have is the plasma membrane here, the cytoplasma here, and you have on the outside here this structure called the glycocalyx, which is important as it will involve cell-to-cell communication and also other various important properties, which we'll get into in a minute. Here, as we've seen the plasma membrane, lipid bilayer, we'll note that there are some other structures inserted into the exterior. This is the outside of the cell. And these are carbohydrates. Some of them are attached to various proteins, thereby forming glycoproteins. And if we move forward, we're going to talk about basically structures for protection, adhesion, and movement. The glycocalyx is a sticky extracellular layer at the outermost edge of the cell. And it's really composed of a diverse collection of carbohydrates, glycoproteins, and glycolipids. It is used for attachment. It is used to protect the cell from stresses and to assist in cellular communication. In multicellular organisms, this is central. A glycocalyx is central for proper tissue development. Okay, so it tells some cells how to develop, how to build up layers, how to know when to stop, where to go, etc. In pathogenic fungi and protozoans, the glycocalyx can be used as a defense mechanism against host immune defenses or to enhance infectivity. Again, that we'll talk more about when we get into host microbe mechanisms. The flagella is a long tail-like structure used in motility. Many gametes and protozoa use flagella. The flagellum is wrapped in a cell membrane covering. The inner part is continuous with the cell cytoplasm. The structural interior of the flagella is made up of the protein tubulin, and they are arranged in a 9+2 arrangement, which I'll show you in a micrograph in a moment. The flagellum is anchored to the cell by the basal body. So this is a cylindrical collection of microtubules, and they sprout actually from the centriole. The flagella move in a wave-like format, back and forth motion. What I encourage you also to do is review the comparison of prokaryotic and eukaryotic flagellum. If you take a look first here, here's the flagellum. You can see the plasma membrane covering it. You can see the microtubule pairs in a 9:2 arrangement. And by the way, this is not empty space in between here. As you can see here also, there is some sort of attachment between the outer tubule pairs, microtubule pairs, and that inner set there. Down here would be the basal body, which sprouts off from the centriole. The wave-like motion of eukaryotic flagella, you'll notice it's sort of a wave back and forth movement. Now, we're going to get into this in another minute about cilia, but I wanted to just give it to you as a perspective. Cilia are usually shorter, and how they move is sort of like oars. They go forward with a power stroke, and then what happens is they collapse and whip back in their recovery stroke. And this is all in a coordinated fashion to direct the cell in one particular direction. So they're all beating in a direction that kind of power strokes down this way, recovery stroke, and keep going to allow it for normal flowing motion. Here's where I want you to be mindful about prokaryotic versus eukaryotic flagella. First off, prokaryote, they're built with flagellin protein. Eukaryote is with tubulin. Prokaryote do not have microtubules, whereas eukaryote does, and it's in a 9+2 arrangement. The membrane is not enclosing the prokaryotic flagella, except for periplasmic flagella. If you remember, those are the ones you see in spirillum bacteria, such as the bacteria that cause syphilis. The eukaryotic flagella are enclosed by a membrane. The anchoring of a prokaryotic flagella is a hook and filament structure anchored by rings. The eukaryote is by microtubules which sprout from the centriole. Now, with the prokaryotic flagella, its motion is rotary, sort of like a propeller, so it's kind of spinning. Whereas with the eukaryote, it moves in a wave-like manner, whipping back and forth, back and forth. With cilia, now we're dealing with, this is Latin for eyelashes. They're structurally similar to flagella, but much shorter and much more numerous. Only eukaryotes really have cilia, and they provide for synchronized rowing motion. Ciliophora protists use it to move around in the water. Cilia are also found in animal cells. For example, you have cilia in the trachea, and this is useful for sweeping away mucus and pathogens away from the lungs. Now, intercellular structures we're going to be focusing in on next. First off, the ribosomes. Now, ribosomes can be freely moving around in the cytoplasm or attached to the endoplasmic reticulum. That's where you get the type of endoplasmic reticulum we call rough endoplasmic reticulum. Now, the ribosomes in eukaryotes are usually two subunits, the larger 60S, the smaller 40S. All ribosomes are composed of proteins and ribosomal RNA. They are essential for protein synthesis. You can't live without them. That's why toxins that target eukaryotic ribosomes are nasty. They basically will lead to the death of the organism. An example of this is ricin, otherwise known as castor bean toxin. This will bind to the 60S subunit and basically shut down protein synthesis. Also, toxins made from Shigella and E.COLI can inactivate the 60S subunit and disrupt protein synthesis in affected cells. Here is our 60S subunit. Here is our 40S subunit, small, large. Bring them together and you've got a complete 80S ribosome. That's going to mean that it's going to be having a sedimentation at 80. Okay? Next we're going to deal with is cytoskeletons. Now, cytoskeletons are an intercellular network of protein fibers. They basically help maintain shape, facilitate movement, protect against external forces that might distort the cell, direct transport of vesicles and organelles and other cellular cargo. So it's sort of like a transportation network as well as a structural system to hold the cell into certain shapes and structure, etc. Now, some pathogens may disrupt the host cell cytoskeleton, and that will help for pathogen invasion. Salmonella enteritica is one. Other pathogens may hijack the cytoskeleton fibers to help propel the pathogenic cell from one host cell to the other. So if you have cells next to each other, for example, Listeria monocytogenes, that will be in one cell, but it will use the rail, the cytoskeleton, as sort of a vehicle to help propel it so that it can smash through the cell membranes and go right into another neighboring cell. Now, the three major fiber types: microtubules, intermediate filaments, and microfilaments. The microtubules are hollow tubules made of the protein tubulin. They will have a diameter of about 25 nanometers. Remember that nano is 1 billionth. So when I say a nanometer, that's 1 billionth of a meter. Intermediate filaments are rope-like fibers. They're about 10 nanometers in diameter, and they can be made from different proteins such as desmin or keratin. Microfilaments. These are very fine fibers. They're made actually up of actin protein. And if you remember your anatomy and physiology, you know about the thin filaments and thick filaments that you find in a sarcomere, in the skeletal muscle. The thin filaments are made of actin microfilaments, and you have myosin as the other thick filament. So actin protein, the microfilaments, are going to be anywhere from 3 to about 6 nanometers in diameter. A lot of times they're associated with the motor protein myosin to facilitate movement. Here is my centrosome and my centrioles and my cytoskeleton elements out. And you can see how they spread out and will basically help reinforce the shape of the cell. Now, I don't expect you to memorize every little component of this, but I do want you to get familiarized with these, especially how we've moved from extracellular now into intracellular. We have to deal with now eukaryotes of their membrane-bound organelles. First off, the nucleus. This is a membrane-bound organelle to store DNA. Now, as you look at the nucleus, you're going to always note that it contains a lot of DNA, but some of the DNA is going to be tightly wound up in the form of chromosomes. Others are going to be loosely wound or unwound. Chromatin is the loosely wound DNA. It has proteins on it. Usually these proteins are histones, but they can be other proteins as well. Nucleoplasm is the liquid inside of the nucleus. The nucleolus is the site of ribosomal subunit assembly. So this area will consist of proteins and RNA. Nuclear envelope. Now this is a double membrane structure that surrounds and protects the DNA, yet it has pores to allow entry and exit of substances. The nucleus is surrounded by the rough endoplasmic reticulum, RER. It's studded with ribosomes. Now, the key point here is that the ribosomes are going to still synthesize proteins, but as they make the protein, they're directly inserting the synthesized proteins right into the inside of the RER. This is going to be involved with packaging and processing. In other words, chemical changes, maybe adding certain sugars to select parts of the protein, etc. Here you see the nucleus. You see the nucleolus here, some of the loosely wound chromatin fibers. You have the liquid nucleoplasm here. You have the double membrane of the nuclear envelope, and you have the nuclear pores here, 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 etc. Now, the nucleus is surrounded by the rough endoplasmic reticulum, which we talked about. RER is studded with ribosomes. We've talked about some of the function of the ribosomes when they are attached to the rough endoplasmic reticulum. Okay? But other structures are equally important. One of them, the smooth endoplasmic reticulum. Yeah, so that's not studded with ribosomes. Yes, that's true. But what smooth endoplasmic reticulum is a specialized membrane-bound structure for chemical processing, lipid synthesis, detoxification of select chemicals, such as alcohol, drugs, metabolic byproducts, etc. When you have a cell that's doing a lot of this detoxification, for example, a hepatocyte, a liver cell, you're going to see gobs and gobs of smooth endoplasmic reticulum present. And that's what you actually do in micrographs with select staining. Now, another membrane-bound structure is the Golgi apparatus. This is a membrane-bound structure that sorts, packages, and ships proteins and lipids around the cell or out of the cell in vesicles. The Golgi apparatus will encapsulate into a lipid vesicle, a lipid membrane vesicle, certain products so that they will be exported out of the cell via exocytosis. Or some of the products may be made into vesicles that will be transported to other parts of the cell. Here we have the rough endoplasmic reticulum. The turquoise little specks there are basically the ribosomes. Here we have these nice smooth endoplasmic reticulum, which are involved with certain biochemical activity. And finally, we have the membrane-bound structures of the Golgi apparatus. And in the spaces, they call that the cisternae. And we have one side that receives these vesicles from the rough endoplasmic reticulum and begins processing them and packaging them in vesicles that may be shipped out or sent to other parts of the cell. Here is another EM, electron micrograph, of the Golgi apparatus. Here you can see the receiving end, and it's almost like there's a processing or distillation or concentration, purification, whatever, that occurs with different products, and eventually they bubble off as vesicles. And that brings us up to vesicles and vacuoles. Okay, what are the different types of vesicles that exist? One is transport vesicles. They basically move substances around the cell. Vesicles destined for the plasma membrane may be involved with replenishing the lipid bilayer and supplying membrane proteins. Also, secretory vessels. These are very special vesicles that transport wastes or other bioactive molecules out of the cell. This is in the process called exocytosis. Now, lysosomes are very special. They are vesicles that contain various hydrolytic enzymes which break down substances engulfed by the cell during phagocytosis and receptor-mediated endocytosis. Then we have another type of vesicle called a peroxisome. Now this is unique. This is a vesicle that contains enzymes that break down fats and amino acids, and it will also protect the cell from hydrogen peroxide and other toxic oxygen intermediates. For example, an oxygen that might be O2 with a negative charge, it might be hydrogen peroxide, but it also has a negative charge because anything that's charged will then chemically react with anything around it. Fatty acids, any type of lipid, DNA, or protein. So you never want to have what they call free radical, oxygen radicals, etc. around. You neutralize them. Now, vacuoles are vastly different because they're a conglomeration or a conglomerate of many vesicles merged to make a larger membranous sac. This can be central in the structure. It could actually take up most of the volume of this, what was then the cytoplasm. The vacuole may contain water or nutrients or waste. It's very common to see vacuoles in plants and fungi. Now, there's an interesting other sidebar here. Some protists, such as paramecium and amoeba, use a structure to manage osmotic pressure to avoid lysing the cell in a hypotonic environment. What does that mean? Well, if you take an amoeba and you put it into fresh water, basically the water outside of the cell is hypotonic compared to what's inside the cell. Inside the cell is going to be somewhat lower in water content, but also higher in solutes. As a result, there's always going to be this osmotic pressure water trying to get inside the cell. But you can't have it just suddenly just rush in and not do anything else, because the cell will fill up and burst. So they have this structure called a contractile vacuole. And what it does, literally, if you look at it under a microscope, you'll see it expand out a little bit and then contract back. Expand out and contract back. And what it's literally doing is taking in excess water and pumping it right out of the cell. Now, we're getting into the last couple of parts here, because we come back to where we almost started in the beginning, the mitochondria. The mitochondria makes most of the cell's ATP. Remember ATP, adenosine triphosphate? This is a double membrane organelle with its own 70S ribosomes and circular chromosome. Gee, just like bacteria. Yeah. It makes amino acids and vitamins. It regulates cell division. It contributes to apoptosis. Now, if you're not familiar with apoptosis, that's programmed cell death. The structure of the mitochondria consists of the intermembrane space, the outer membrane, the inner membrane. Between the outer membrane and the inner membrane, yes, is the intermembrane space. We'll get into that. And you also have a large surface area of the inner membrane. And as a result, you also have these cristae, which basically this is going to lead to an incredible amount of ATP produced, because in that membrane will also be ATP synthase. When we get into cell energetics and we get into metabolism, we'll talk a little bit more about that. Now, the number of mitochondria differ in different cells. Cells that need a lot of energy, let's say skeletal muscle cell, they're going to have a lot more mitochondria. Cells that don't need a lot of energy or don't do a lot of energy, like an adipose cell, fat cell, they're going to have very little mitochondria. But yet you will find in eukaryotic kingdoms, every kingdom has mitochondria. Even plants have mitochondria. Here is our intermembrane space, the outer membrane, the inner membrane. And you can see how these inner membranes have folds, etc., to increase their surface area. And you have these areas such as the matrix. This is going to be loaded with enzymes that will play a role in the Krebs cycle. You will also have embedded into this membrane certain components of the electron transport chain. And all of this leads to the production of an incredible amount of ATP. Now, the other cell that we talked about a little bit in the beginning, and now we're going to come down to, is the chloroplast. Chloroplasts allow cells to harvest energy from light. And basically they do this by using light-collecting pigments, chlorophylls and carotenoids. You've not heard about carotenoids. These are the pigments that you see, such as the strong orange color or the strong yellow colors that you see, things like in carrots and squash and things like that. Now, inside of chloroplasts, you have this inner fluid site called the stroma. And it contains intermembrane structures that are loaded with pigments. And these structures are called thylakoids. When they're stacked in structures, they're called granum. So think about it like coins. You have a stack of them. An individual coin would be called a thylakoid. A stack of them would be called a granum. Now, inside of the cell chloroplast membrane, there's two membranes, an outer and an inner. The inner, inside of it, has all of these thylakoids. When energy is captured by sunlight, and it is used to incorporate carbon dioxide into sugars. So chloroplasts will take the process, without getting too technical, without getting too much into it, you basically capture the energy, make it into first ATP, then you make it and use it to incorporate carbon dioxide, fuse them together to make sugars. Sugars, in essence, are going to be your storage, your energy to be stored later on to use. How are you going to use it? By glycolysis in the cytoplasm, and then by the other two steps, the Krebs cycle and the electron transport chain that are in the mitochondria. Here is our outer membrane of a chloroplast. Here is the inner membrane. In this fluid-filled space, it's called the stroma. Here is an individual thylakoid, which has a lot of pigments embedded into it. A stack of them are called a granum, and a connection between one granum and another is called a lamella. Well, that's about it for this introduction to eukaryotes. You want to review the visual summary in your textbook on page 115, review the chapter overview on pages 116, and review the end of the chapter questions, pages 118 to 119. Also, in Mastering Microbiology, you have to review a homework assignment which is due by midnight or 11:59 Friday. And you have the quiz that's in Mastering Microbiology, which is due 11:59 on Saturday. You also want to take time to peruse in Blackboard the lecture videos that I use to help support some of these concepts and help you to review. Now, our next section will be on genetics, and that will be chapter 5. Have a wonderful day.